Button. Okay, hooray.
Hooray!
Hooray. Okay,
Hooray.
so we're going to explain these things to you.
Homeostasis.
Homeostasis, and then I'll talk about feedback loops, which is how our body maintains homeostasis. And then I'll talk more about proteins, which is how the body then affects us staying within range of homeostasis. Really interesting.
But what's homeostasis?
Thank you. I was about to explain that. Homeostasis is your body trying to stay within some kind of set range of equilibrium. So I'll draw a graph here. We like graphs in physiology. So we'll talk about, say, body temperature. Body temp. You can read that sideways?
I can.
Great, okay. And then this is time. No units, doesn't matter. This is just some amount of time.
Okay.
So average body temperature is like 37. This is in degrees Celsius. That's what that means.
Reasonable.
So 37, and then say you can go down as like 35 degrees, and then 39 degrees. So average body temperature is this 37, but actual body temperature will fluctuate throughout the day. So I'm just going to draw these lines. So the range that we like to stay in is this range. This is where our body temperature likes to stay in. But the average, it is always trying to stay at what we call the set point, which is like, say, right here. So there's set point, try to stay within this range, and that's what homeostasis is.
Right.
And so I'm going to use this red line. This is going to be your body temperature, say, like throughout the day. So like, start off here, and then you get warmer, and then you get colder, and then you get warmer, you get colder, and you get warmer, and so on.
Makes sense.
So with homeostasis, you can go above or below the set point, but you're always staying within this range because this is the optimal body temperature for the example that your body is trying to stay at.
Okay.
So then what becomes important is how the body checks this stuff. So the next thing is like the pathway that your body does to check if you're within homeostasis, and then how it will change and make differences on that. So it starts with, I'm going to use a different color. We'll use green, because green is good. Good for green.
Green is a creative color.
Okay, is this still in the thing? Okay, it is. Okay, so it starts off with some kind of receptor. For example, with body temperature, these are like temperature sensors in your arm. And so they'll what's the word? Basically, they're just going to check and say, they'll measure what it is. So, measure.
What, for example, my body temperature is?
Yes.
Oh, okay.
So, they'll measure some variable. In our example, the variable is body temperature. So, this receptor will then will give this information to what's called an afferent pathway. Afferent means from peripheral to what we call the control center, which is usually the brain. And this, done by neurons or hormones. So it will either use nerves that will then send information, or it will use the blood to circulate hormones, which will then also get to the brain or the control center. So that will then send information to, I'm going to do a capital L, control center, or central command. And so most often this is your brain, but other times it can be places within your spinal cord, basically anything that's within your central nervous system, because those things know what your range and what your set point is. And so it's like, all right, I know what it is, so then it uses what's called an efferent pathway.
Ooh.
Efferent, which means from control center to the periphery, which will then go back to some other receptors to make some kind of change on that variable.
What's a receptor?
I'm not there yet.
Oh, okay.
I'll talk about that. Those are the proteins. So say, for example, you've got some kind of temperature sensing nerve in your body, which will then go from that nerve through the afferent pathway to your brain, and your brain says, are we within our set point or not? And so say you're like here, it's like, that's a little warm. And so control sensor says, we're too high. Efferent pathway, it will then send signals through hormones or nerves to glands in your skin to make you sweat, which will cool you off. And that would be send signal, receptors say, we received this information, we're going to sweat.
Okay.
Which will then bring your temperature down.
And then once it gets there, the cycle repeats.
If you go down, then the body, say you're like, it's really cold outside and you're shivering. They're measuring your body temperature, says, we're really cold. Goes through afferent pathway to control center says, we're too cold. Then using the efferent pathway, it sends signals to your muscles, makes you shiver, warms you up, brings you up.
I see, very cool.
Yes, so those are examples of what we'll call feedback loops. So I'm going to erase this.
What kind of things can disrupt homeostasis?
Actually, I'll get to that. That's a really good question when we talk about different kinds of feedback loops.
Okay.
Good question. So there are two kinds of feedback loops. We have negative. I feel like Khan Academy. So we have negative feedback loops, and then we also have positive feedback loops. Positive. We're going to talk about negative. So a negative feedback loop is brings variable towards set point. Set point.
Which is that central blue line, if
yes.
I remember right? Okay.
So if we're talking about the body temperature, so if it's too hot and you're sweating, that's a negative feedback loop trying to bring it back down towards that set point.
Okay.
And then if you're too cold, you shiver, brings you back up. So that'd be a negative feedback loop. Negative
Both
is...
of those would be negative feedback.
Yeah, so it's your body trying to keep you within that set point if you go too far away. So say you're like in really, really, really cold temperatures and you're starting to get hypothermia and you're going way, way down here. And then you start shivering a whole lot and you put layers on. Then, you'll slowly start coming back in. So even though at the beginning you were going even further away, once you start going back in, that's still a negative feedback loop.
Okay.
Always, if it's going back in towards a set point, always negative feedback.
Right.
Okay. Now we're going to talk about positive feedback loops. These are really interesting.
Okay.
But understand negative feedback loop?
I do.
Great. Okay. Positive feedback loops, we have two kinds that we're going to talk about. So we have intentional, and then we have what's called inadequate negative response, which is the more interesting thing, which is what will inadequate negative, and then I will explain why it's called. Actually, I'll just inadequate negative response. Okay, so intentional positive feedback loops are things that happen. I'm going to erase some of these lines, and I'm going to make this no longer about body temperature.
Okay.
Because body temperature is something we usually try to keep within a nice little set range. I'm going to make this, the best example that I can think of is amount of oxytocin. Actually, I'll just explain this one. So when a woman is extremely pregnant and about to go into labor, the baby's head will push on the cervix, which releases oxytocin in the brain. So we have beginning, say here, no we'll just say some odd, regular amount of oxytocin. as the baby's head pushes on the cervix, oxytocin goes up. As oxytocin goes up, the brain sends signals down to the abdominal area in the uterus, which says, go into contractions and try to keep pushing the baby out. So as that pushes the baby more, the baby's head pushes more against the cervix, which releases more oxytocin, which then tells the brain, push more. And so it's this constant loop of pushing more and more as more oxytocin gets produced.
Feedback loop, you might say.
Yeah, positive feedback loop.
I see.
So it drives it further away instead of getting closer. That makes sense?
Yes.
Okay. So that is a good example of an intentional positive feedback loop, because the body is intentionally trying to do this.
Sure.
Your inadequate negative response. Example I really like is we're going to talk about blood pressure. Conveniently writing in red. Okay, so blood pressure. So there are things that your body uses to, I didn't talk about that in there. Okay, the afferent pathway uses what's called effectors. So effectors are things that change that variable. So like how I wrote receptors at the end, those are your effectors. So like your muscles are effectors to make you shiver, your sweat glands, those are effectors to make you sweat, bring you within that homeostasis.
Okay, yeah. Easy enough.
So blood pressure, the effectors that we have are blood volume. Blood volume is the, what's the word? I don't remember. I'll remember it in a second.
Okay.
But blood volume is not an effector. The effectors that we want to care about is heart rate. I'm going to write effectors here. Effectors. So we have heart rate. So how fast your heart beats. If your heart beats faster, you have more blood pressure.
Right.
And then we also have what's called vasoconstriction slash vasodilation. That is your arteries either constricting more, which increases blood pressure, or decreasing and not constricting as much and dilating, which reduces blood pressure.
Right.
So we have our set point. Our body wants to stay somewhere within this range. If you get a cut on your arm and your blood volume, okay, I'm going to start marking these in different colors. I have 4 colors. That's what I want. Blood pressure will be in red. Blood pressure is red. Heart rate will be in green. Vasoconstriction and dilation will be in blue. And then blood volume will be in purple.
Perfect.
And all of those will be on this graph.
Right.
Okay. So say you get into a serious car accident and you have a pretty intense gash on your leg.
Oh no.
Your blood volume starts dropping. This is not great, because if you don't have the right amount of blood, you don't get nutrients to other places within your body that you need.
Sure.
Really bad. You bleed out, you die. This is a no bueno. What happens is, as this falls, your blood pressure then immediately starts to drop because you have less stuff. So blood pressure starts to drop. Then what happens is you have receptors that can test like the constriction and dilation, so the pressure on your arteries, and they say, we're like high blood pressure, low blood pressure, and sends signals to your control center somewhere in the brain, I would assume. We haven't quite learned about that pathway yet. Well, the exact pathway. So that sends signals, and then your brain will cause either of these two things, usually both.
Okay.
So say this is normal, and then we start losing blood, and then heart rate is going to start going up. as
To try to increase blood pressure.
Pressure.
Right. Okay.
As will vasoconstriction. So this will also go up. So if this were to continue and then your blood pressure would goes down and then starts going back up, that would be negative feedback loop. That's what we want.
Right.
But if you lose way too much blood, your blood pressure can continue dropping, and even though these are going up, so you vasoconstrict, so you keep trying to go up, and your heart rate keeps pumping faster. Well, your heart keeps pumping faster, but because these are going up, that actually pushes your blood out more if it's a large enough wound.
That makes sense.
Which ends up causing there to be less blood, which means less nutrients are getting to the heart and to the brain, which actually ends up causing your heart, green marker, to start pumping less.
Oh no.
Because your cells start dying because they don't have the right nutrients, which means your arteries don't constrict as much. And then those start going down, and then because these are going down, your blood pressure starts going down even more. And then because your blood pressure is going down, you don't have enough nutrients going to your heart keeps going even slower, and then this keeps going even slower, and ends up turning into a positive feedback loop because you've lost too much blood, and your blood pressure drops, and then you just die.
I see.
So this is an example of the inadequate negative response, because at the beginning it tried, but because I had lost so much volume, it didn't really make a difference. So it ended up actually making it worse.
Okay. Dang. That makes sense though.
Okay. Any questions?
No.
Great. Okay.
All right. I have a question.
Yes.
So, so are the intentional positive feedback loops good
Yes.
all the time?
Yes. So they're called intentional.
Okay.
So those are controlled by control center, and it tells the body to do this specific thing.
Okay.
Baby pushing its head on the cervix like birth, that's the main one that I can think of when I think about intentional positive feedback loops. I can't
But
think
for
of
the most part, it's your body intentionally doing something.
Yeah, intentionally trying to drive you further from
I see.
the set point.
Cool.
I don't know why I put that in quotation marks. Further from the set point. Okay, that's homeostasis. Now we're going to talk about proteinins.
Ooh, I can find out what receptors are.
Yeah.
Exciting.
These are great. Okay. This is a cell membrane. Pretend.
Yeah.
Great. So we have these fun labels. ICF and ECF. ECF stands for extracellular fluid.
Right.
So it's the water that is outside of the cell.
Okay.
ICF is intracellular fluid, so it's stuff inside the cell.
Okay.
Great. No confusion?
No, I think I remember learning something about this, so yeah.
Okay, so we're going to talk about the 4 different kinds of stuff that I like. So your cell membrane is made up of a phospholipid bilayer. So what that means is you have a hydrophilic head, and then hydrophobic tail. And so you've got 2 of those. So you've got the head that, H2O with a heart because it likes water.
Right, yeah, okay.
And then it has this is a phosphate head, which is polar, which means it likes other molecules that have kind of like a partial charge. The tails are made up of a bunch of carbon and hydrogen, which are non-polar. So H2O, no likey.
Okay, yeah.
And then your cell membrane is one of those, and then another one,
But the other way around.
but upside down. So what this prevents is it doesn't prevent anything that is charged to make it through very easily, or anything that is polar. And what polar is, like, electrons kind of move around the molecule and aren't like equally dispersed around it.
Oh, okay.
So small things like carbon dioxide and oxygen are small non-polar molecules, so they can diffuse no problem through this. Big things, or like say like certain kinds of like proteins that you eat, or stuff like glucose or sugars, because they're large and they're polar, they can't make it through the membrane as easily. So your cell has to have some kind of way that it can move stuff in and out. Otherwise it just dies and gets filled up with its own waste, or doesn't get enough food and just dies anyway.
Okay, yeah, makes sense.
Which is why proteins are super important.
Cool, so that's what that line is made of.
Yes, that's what this is. This is our bilayer.
Gotcha.
But it's really easy to just draw a single line.
Yeah.
So the first protein channel that we're going to talk about is literally just called a pore. So it's just this little hole in the membrane, and it just lets stuff go in and out.
Okay, yeah.
So a good example for this is what's called an aquaporin. So it lets water move in and out freely within the cell.
Gotcha.
So H2O, it can go in or out. So aquaporin is just a hole in the membrane. Cool. Next thing we're going to talk about is mechanical gated channels. It's a little hard to draw like a three-dimensional structure. Okay, we're going to just draw this little block. So a mechanically gated channel or protein is, so your touch sensors in your arm are mechanical. So if there's any kind of stretching in the membrane, it will cause this to open. And then it will let something come in.
If they're stretching.
Yeah, if the receptor, if the protein itself literally stretches open. So say I've got like a bunch of glucose. I'm just gonna write sugar. So I've got a bunch of sugar out here, and this is a mechanically gated channel. If something stretches, it lets the sugar come in, or potentially lets something go out.
Okay.
The next one is ligand gated channels. I already used blue. Let's use purple. Okay, and then I'm going to put a little cover on this. The ligand gated channel, that's also purple. I'm going to draw a little indent on this. So a ligand gated channel does not open unless what's called a ligand, so some kind, basically like a key to a door. If the door's locked, you can't open the door until you unlock it. And so ligand gated channels, if I have some kind of chemical bind here, it causes the protein to change shape. In this example, it causes it to open, which then allows allows ions to move freely through the protein channel.
Okay.
So, but if this doesn't bind, then it just stays closed. Draw those. Otherwise it stays closed. So it's kind of like the key to a door.
So like maybe a protein with the right shape or something?
So the place where it binds is called the allosteric site. So specific chemicals will bind to specific allosteric sites. So if it doesn't match the keyhole, like if I'm trying, say the key to my car to the key to our apartment door, it's not going to work. It's the wrong key. So it has to have the specific kind of chemical that will bind to this to allow it to actually open.
Legendary.
This is also how certain kinds of drugs work, because they mimic the natural key to the protein, and they will cause it to either never open or just like stay open. And they may potentially bind for way longer than the actual ligand should. Okay.
That would be bad.
In some cases, yes. For example, one of the ones that we talk about is, I don't remember how it's pronounced, but it's like Weabane. Here, I'll write it out. It's really weird. If I can remember.
Benzomethylecogniine?
No. I think that's how it's spelled.
I see.
Yeah. which is I'm not the most confident how to actually pronounce this, but what this one does is it can, if I remember correctly, it will bind to certain channels and then just keep them open. So what it could do is it could say if it binds to some of the receptors within your heart, it can cause it to contract and then just stay contracted. And then if they're for too long, you just die. Because if your heart isn't pumping, you're not getting blood, you don't get oxygen, you don't get nutrients.
Yeah, it
and
basically just breaks your heart.
Mm-hmm.
Okay. Reasonable.
I don't remember if that's exactly what that drug does, but for the example, makes sense.
Yeah.
Okay, and then the last one is what's called a voltage-gated channel.
Ooh.
These ones are really exciting. The one that I'm going to talk about is our... So this, I'll talk about this one later. Voltage gated Na. That means sodium. Voltage gated sodium channel.
Right.
So for your average neuron, which is where I will generally talk about these more, at least right now, you have a membrane voltage of negative 70 millivolts. Basically what that means is the inside of this cell has more negative charge than the outside. A millivolt is a volt is a separation of charge. So outside is more positive than the inside, making it minus 70 millivolts. And this is what the protein is used to. If this changes, say I end up getting a higher negative on the outside and then a positive on the inside, this actually will just open. This will open and it will allow our little sodium ions to come into the cell.
Which would make it more negative.
Yeah, it would make the outside even more negative.
Oh.
But this being voltage gated makes sense. It literally only opens if some kind of charge difference happens.
Oh, so it doesn't really necessarily matter what side has more charge. It's just
if it changes, it opens.
Oh, okay. Okay, yeah. Okay, cool.
Okay. Great. Oh, another thing that we're going to talk about. Specificity and rates. So these channels, actually ones like this, are super important. So you have multiple of these voltage gated channels. I'm going to draw another one. This one I'm going to do in green. So the two main positive ions that your cell uses are potassium and sodium. Potassium is Ka plus, and then sodium. So potassium, because it is further down on the periodic table, means it is a bigger ion.
Okay.
So you might think, oh, what prevents sodium from going through this? So the super exciting thing is what's called a hydration sphere. So because these are positively charged ions, and water is a polar molecule, so it has a partial negative and a partial positive somewhere within the molecule.
Sure.
That's more chemistry stuff, and it's exciting. So either way, there's our H2O molecule. The oxygen is more negatively charged because oxygen is a mean grabby atom. So you end up getting a whole bunch of waters surrounding the big ion. And these channels have a bunch of charges on the side that will strip the water off of the ion and allow just the ion to move into the cell.
Okay.
The problem with sodium is it's still got those. I'm just going to draw that. But because it doesn't exactly match these, those aren't able to strip the water off, so it hits it and kind of just bounces off.
Oh, okay. Yeah. So it absolutely must go through the voltage gated sodium channel.
Yeah, so that's when we talk about like specificity. Proteins will only allow specific things to go through them,
Mm.
which is why we have so many different proteins in our cell membrane.
Nice.
So like we'll have ones where we talk about stuff that's specific for sugar and will only let sugar in, and then ones that will only let potassium in, ones that will only let sodium in. So that's the specificity. And then there are other channels where it's just basically just a big colon will let literally anything in.
Like the H2O channel.
Well, that's specific for water.
Right. Okay.
But they're like within our neurons, so the junction between the neurons called the synapse, there are some nerve connections where they will literally just let basically everything flow through.
Mm.
From inside one neuron to into the next neuron. Doesn't matter what it is, they're non-specific and they just let everything go through.
Okay.
Okay. Okay. I don't know why I erased that, but
Cool.
I'll redraw those. Any questions?
I don't think so.
Okay. Other things with proteins is the more proteins you have, the quicker something can make it across a membrane. And then there's a couple other things. There's an equation called Fick's law of diffusion that determines how quickly something can move across a membrane through proteins or not. So one of those things is what's called the permeability, which deals with whether or not the membrane is permeable, so if it can pass through, or how many proteins there are. So if I add a bunch of proteins, which I'll draw out our membrane, if each of these lines is a protein. So if I add a bunch of these compared to something that only has 3 proteins, which one's going to be able to move more stuff faster? The one with more proteins, right?
Okay. Why?
Because I have more of these that can move stuff.
Oh. Okay, yeah.
So if I have more proteins, I can move stuff faster in. So if I have 100 children outside of a classroom and there's only 3 doors, they're not going to go in nearly as fast as if I have the same amount of kids, but I've got 7 doors that they can go into.
Okay, yeah.
So the more doors I have, the quicker they can go in.
Okay, so yeah, those are like the gates or whatever.
Yeah.
Cool.
Another thing that changes is the thickness of the membrane. Okay, I'll just draw that. So I'm going to draw a super thick membrane versus a really, really thin membrane. So if I've got some kind of really big molecule, say that, which one do you think it's going to make it through faster?
Probably the thin one.
The really thin one, yes. The thinner the membrane, the quicker something can diffuse across, which is also why our cells are around all the same proximate size, pretty small. I think it's 1 micrometer cubed, approximately, because it's a good size for molecules to diffuse across and get to where they need to and provide the cell, because the bigger you are, actually that's the next thing we're going to talk about. Talk about area. This is going to be like a flat plane. If this is one side of my cell, that's still in the picture, right? Yeah, okay. So if this is one side of my cell and it's this little box, I can't get as much diffusion into this as I could if it's got more space.
Because there's more
area.
Yeah, okay.
I have more space for stuff to just kind of go through. Like if I have a bunch of oxygens that I'm trying to push through, it can go faster when it's got more area rather than a smaller area.
Yeah.
So this, what happens is our cells have a good balance of large area but also thin membrane. Because if you keep increasing the thickness of the membrane, you have to increase the area of this. But if it's too thin, really small area, it diffuses fast, but you don't get as much as you would want.
Yeah, okay.
So you end up getting a large area, but also not a super thick membrane, and that's about the area that our cells are at. So they're the bigger area, you can move stuff through faster. thinner, you can move stuff through faster. Makes
Yeah.
sense?
Yeah, no, that's reasonable.
Okay. Specificity rates that. I think that's it for Fick's law. Talk about all of those things. Okay. The next thing we're going to talk about is different types of transport.
I have a question.
Yes,
What's a receptor?
those proteins.
Oh, okay. Okay, yeah, cool. Okay, transports.
Okay. So when I say receptor, I'm basically just meaning receptor protein.
Oh, yeah.
So something that detects some kind of change.
The doors for the children.
Yeah.
Alright, cool.
Be kind of like if there's a teacher standing on the outside of the door and seeing, children, and then opens the door.
Nice.
Receptor, it says.
Okay, yeah.
Sees something, changes. Sees. Quote unquote.
Yeah, right.
Okay. Okay, so we've got passive transport. I'm going to draw a big tree. So we have active and passive. How long have I gone for? 36 minutes. Oh my gosh. That's okay. Okay. Active and passive. Passive transport is transport across the membrane that doesn't require energy. So this is through the use of concentration gradients. In chemistry, we'll talk about sodium. So really big means there's a whole lot on the outside. ECF, then ICF.
Cell membrane.
Yep. And then I'm going to draw a little one, implying that there's not a lot on the inside.
Okay.
For your cells, actually for basically anything, if there's some kind of difference in the amounts, they want to make it equal.
Yeah.
So if you've ever dropped like a droplet of water into, food coloring into a glass of water, over time it spreads to the whole thing and then it's all the same uniform color.
Right, it wants to reach
We call it
equilibrium.
equilibrium. Yeah, good.
Yeah.
So this wants to go this way.
Right.
So if I have some kind of protein right here that allows the sodium to go through, just of its own accord, eventually it will diffuse and then it will reach equal concentrations. That's an example of passive transport.
Okay.
Just going through, reaching its equilibrium state.
Cool.
Doesn't use energy. Great, we like it. And then there are different types of passive transport that there's protein-mediated transport. There's also what's called exocytosis, which is basically where you have the membrane and then there are our receptor proteins. I'm just going to draw them like that.
Why? Get it?
Yes.
Okay.
I get it. And say we've got some kind of, say this is just like sugar, and then it will attach to this, and then basically it just forms a little bubble of membrane that moves inside the cell. Really interesting.
Yeah, okay.
So that is called exocytosis. The cell pulling things into it.
Sure.
Yeah. Okay. There's your passive transport. Uses those. Active transport is something that requires energy.
So like your heart pumping blood through your body or something?
Not quite.
Okay.
So passive uses the energy of the concentration gradient. It just goes down the gradient.
Right. Yeah.
Moving from high to low. Active transport is doing the opposite. So for potassium, we have a large amount inside our cells and not a lot outside our cell. So active transport we have, so we've got some kind of protein channel that pumps potassium from outside into the cell.
So that there's even more of a difference.
Yeah, so you're increasing that gradient. And this uses a molecule that we call ATP, which is the...
Well, I've heard of that one.
Yeah, so that's our general energy molecule within the cell. So it uses this, turns it into adenosine diphosphate. Yeah, ADP. So this is adenosine triphosphate, adenosine diphosphate. So it removes a phosphate group.
Right.
And by doing that, it creates energy to move the potassium into the cell.
Cool.
Increasing concentration gradient. So that is our primary active transport. And then we also have secondary active transport. Secondary active transport doesn't directly use ATP. This one's really interesting. So, good example for this one. We have, again, we're going to talk about ion concentrations inside and outside the cell.
Okay.
Yeah. ECF, ICF.
Cell memory.
Yeah. So, calcium has a high concentration outside the cell, very small inside. And then sodium has a high concentration and low concentration inside the cell.
Okay.
And then we have some kind of protein. And what this one does is it allows sodium to move down its concentration gradient, which doesn't require any energy.
Okay.
But what then that does is that moves calcium outside the cell.
Why?
So the way that this one works is sodium will bind to this protein and allow it to move into the cell. And once the sodium unbinds to this, it causes a change to allow calcium to bind to this protein. And then once calcium binds, the protein changes shape, and now the calcium is on the other side and is released outside of the cell. We call those conformational changes, the protein literally changing shape depending on what's bound to it. So sodium binds to it, sodium moves into the inside, and then sodium unbinds, which then causes this to change shape, and the calcium binds there. Once the calcium binds there, it moves and then ends up going outside.
Ew, okay.
So it uses the energy of this concentration gradient to push this thing against its concentration gradient.
That's cool.
Yeah.
That's pretty convenient.
So that's your secondary active transport, because something is still moving against its gradient, but it's not directly using ATP.
Okay. It's just using the energy of something else.
Mm-hmm.
Cool.
And so this specific example is called antiport. Antiport is they're moving in opposite directions. One goes in, one goes out.
Okay.
Another one, this one's really interesting. We'll have sodium, chlorine, and potassium. Sodium and chlorine have high concentrations outside the cell. And so what this one does is it moves sodium in, moves chlorine in, and then also moves potassium in, even though potassium has a high concentration inside the cell. And so this is what we call symport, because you have the stuff moving.
What was the other one called?
Antiport.
Antiport and symport, okay.
Yeah, so one goes in, one goes out. Symport is meaning they're all going in, or they could all be going out.
Oh, okay.
So this one ends up using the concentration gradients of sodium and chlorine to pull the potassium in against its gradient.
Ah, all right. Dang. Very cool.
And that's it.
Nice. Yeah. Okay.
Any questions?
No, I don't think so.
Great.
I understand everything that there has to do about homeostasis and all that stuff and proteins.
Great.
I could be a professor now.
You could.